Hello everyone, welcome to our online lecture. I am Dr. Archana Chaube, Assistant Professor, Faculty of Science from the Bhopal School of Social Sciences. Today, we'll do an important topic from organic chemistry. We know when we talk about organic reactions, there are various types of reactions that occur. If we classify them, like for example, substitution reaction, addition reaction, elimination reaction, displacement reaction. So, in today's lecture, we will begin with the first category, that is substitution reaction. Today, I will be discussing the following topics: substitution reaction, the meaning of this reaction, the various types of reactions, the nature of the reaction, reacting species or the attacking species, and the specific type SN1 and SN2 reaction. So, let us begin. Substitution reaction, as the word suggests, substitution means to substitute an already existing group in the molecule. Like for example, in place of A, when A gets replaced, B comes and attaches. This is known as substitution reaction. So, substitution reaction is a type of reaction where a part of the molecule is removed and replaced with something else. If we basically talk about alkanes, alkanes randomly and readily undergo substitution reaction where the substitution of halogen occurs, resulting in the formation of haloalkanes. When we talk about these substituting species, on the basis of these substituting species, the type of substitution reaction can be classified as electrophilic substitution reaction and nucleophilic substitution reaction. Now, as I have said the term electrophilic and nucleophilic, so we already know, we are familiar with electrophiles and nucleophiles. So, what are these electrophiles and nucleophiles? If we break the word electrophile, electro and philes, the word philic comes from the Greek letter which means liking or loving. So, on the basis of that, electrophiles are those species which are electron loving. They are electron liking, and as we know, opposite charges attract each other. So, which species will be liking a electron? Obviously, it will be a positive species. So, if we talk about the definition of electrophiles, electrophiles are electron deficient species. Those species which are positively charged, therefore, they are in seek of electron and they go and attack on those points where there is lot of electron density. So, if we take the example of electrophiles, they will be carbocations and carbonyl compounds. On the other hand, the term comes nucleophiles. So, again the word philic suggests us that nucleophiles are nucleus loving species. So, that means those which like a positive, and we know that nucleus in an atom is a positive center. So, those which like a nuclei, they itself will be negatively charged species. So, nucleophiles are electron rich species, and examples are carbanion, water, ammonia. Like water, it is a neutral molecule, but it has an oxygen which has lone pair of electron. So, any those species which has the ability to donate an electron, either a negatively charged species or a neutral molecule which has an excess lone pair electron to be donated, will be termed as a nucleophile. So, on this basis, we have classified the substitution reaction as electrophilic and nucleophilic substitution. If we further talk about the subcategorization of nucleophilic substitution reaction, you can see that nucleophilic substitution reaction can be broadly classified into two categories: aliphatic nucleophilic substitution reaction and aromatic nucleophilic substitution reaction. The term itself suggests that aliphatic species, when they undergo nucleophilic substitution reaction, and when this reaction occurs in aromatic compound. So, the nucleophilic substitution reaction in aliphatic species is further subcategorized into two types: SN1 and SN2 reaction, whereas the aromatic nucleophilic substitution reaction has broadly the 5 categories: SN1, SN2. Apart from that, it has benzene mechanism, SNAR addition elimination mechanism, and free radical mechanisms. So, these are the different types of substitution reaction that occur in the molecule. Now let us talk about the aliphatic nucleophilic substitution reaction. As I have already said that it is basically of two types, SN1 and SN2. What does this SN1 term means? If we go into the meaning of these alphabets, the S in this means substitution, N represents the nucleophilic. So SN means nucleophilic substitution, whether you are talking about SN1 or SN2. And what does this superscript 1 and 2 represent? The number 1 represent unimolecular substitution and the number 2 represents bimolecular substitution. Now you will ask, what is the meaning of unimolecular and bimolecular? Uni means 1 and bi means 2. So in unimolecular substitution, is it only one molecule that is taking part in the reaction, and in bimolecule is it 2? No, in both the reactions a reaction will be taking place only if there are 2 molecules, because we are talking about substitution of one group with another. So there has to be 2 molecules. Then why we are calling this category as unimolecular substitution reaction? So let us move further and try to understand what is the meaning of unimolecular substitution reaction. See, when we talk about any reaction, these reactions they occur in many steps, and generally when we say an SN1 reaction, it occurs in 2 steps. But since we know the kinetics of the reaction, and when we talk about kinetics, we talk about the slow steps and the fast steps, and in any reaction the rate of the reaction is determined with the slowest step. The slowest step is one of the any reaction is one of the most important reaction. So when we are saying the SN1 reaction, it occurs in 2 steps. The first step, which is the rate determining step, is the slowest step. And if you see the mechanism, what happens if this is any species in which C is attached with X, Y, Z and L is a group. L represents the leaving group, the group which will be substituted, and X, Y, Z can be any species. So this leaving group, in the first step, the leaving group will break, the bond over here will break and it will move away. We know that carbon is a tetravalent atom, that means it has the ability to form 4 bonds, and if this leaving group is going away from carbon and it goes as a negative species. So since this L is going as a negative species, it is taking away the electrons with itself, due to which the remaining state of carbon which is left is having a positive charge on it, and it now becomes trivalent. That means 3 atoms X, Y, Z are attached to it and the 4th space is vacant, and this species, which is a positively charged species, is known as a carbocation. And we know, when we have studied about the reactivity and the stability of carbocation, so carbocations can be of various types, like for example a tertiary carbocation. This is a tertiary carbocation. The number of alkyl groups will determine the nature of the carbocation. This is a secondary carbocation and this is a primary carbocation. So when we talk about the stability, the tertiary carbocation, because it has 3 alkyl radicals attached to it and they have the plus I effect. So the more positive inductive groups attached to a positive center, the inductive plus I groups, they are diverting the electrons towards the carbon, due to which this positive charge, this deficiency on the carbon is stabilized. They try to give the need, or they try to fulfill the need of this carbon and make it more stable. Therefore, the tertiary carbocation, which has 3 alkyl radical, is more stabler. Then comes the secondary, which has 2 alkyl groups, and the primary, and the least is the methyl group, methyl carbocation, which has none of the alkyl radicals. So since a tertiary carbocation is very stable, so all those species, like I had written in the earlier slide, the example of SN1 is hydrolysis of tertiary butyl bromide. any alkyl radical which will be of the tertiary nature will prefer SN1 mechanism, whereas the lower members will generally prefer the SN2 mechanism. So, coming back over here, the removal of the leaving group results in formation of carbocation. Now, this carbocation which is formed, this whole process is a very slow process, and we have seen that during this step only one of the reacting species, that is the alkane, through which the leaving group is coming out, is only involved. The second species is not in the picture, and the concentration of this reactant will only affect the overall reaction. Therefore, it is a unimolecular. The rate determining step involves only one step. Now, the second step is a very fast step. It is the attack of the nucleophile. So, the nucleophile that comes, it will now go, because it is a negatively charged species, it will go and attack on the positive center of the carbon, because it is a carbocation. So, now this entering species has two sides, because this carbocation, it has a planar trigonal geometry. So, because of this planar trigonal geometry, it is a planar structure, just like a equilateral triangle. It is planar, and carbon is in the center with three groups attached to it. So, the entering group can enter either from the left or from the right, because both are equal, and the resulting products will be A and B. Like if it enters from the arrow which is marked as red, we get the product A, and if it is entering from the side which is marked with blue, we get the product B. But if you go and see the initial reactant, you can see that the leaving group was present on the left hand side, and the products that are obtained are, in one, the nucleophile is in the same orientation as it was in the leaving group, the position was the same, and in the other one it has just flipped to the other side, which means that since the entering nucleophile can enter from two different sides, one of the condition is it maintains the same stereochemistry, and it is known as retention of the product, and one of the product is just the opposite. Like if you see an umbrella, this is an umbrella, and the umbrella is having its end, and in one of the product it just becomes opposite, it just flips to the other side. So, inversion of the product has occurred, because it is a if we talk about the percentage of the product, it is just a chance of probability that the entering nucleophile can enter either from the left or from the right. So, therefore, 50% chances is the product to show the same orientation and show retention, whereas 50% chances for the inversion. So, therefore, this is one of the important characteristic of SN1 reactions, that the product which is obtained is 50% retention and 50% inversion, and the rate determining step is affected by only the concentration of the main reactant alkane, and it is not affected by the concentration of the nucleophile. So, this is all about SN1 reaction. In the next lecture, we will be discussing in detail the SN2 mechanism. I hope you must have understood the basics of substitution reaction and SN1 mechanism. Thank you.